Hi everyone, today we're going to be talking about MCAT carbohydrate metabolism. Okay, starting off with glycolysis, it is important to note that while all body cells do carry out glycolysis, in cells like red blood cells, glycolysis is the only metabolic pathway that they perform since they don't have mitochondria. We can also note that the pathway of glycolysis will produce certain intermediates that can be used in other metabolic pathways, such as fatty acid creation in the liver. To break down the process of glycolysis, we will be mainly focusing on enzymes that are the most highly tested on the MCAT, those that are important, they have important energetic functions, or those that are highly regulated. The first thing that will happen before glycolysis can occur is that glucose will be transported into the cell and phosphorylated by a kinase called glucokinase to prevent it from being transported back out of the cell. The reason that this works is because glucose transporters are enzymatically specific to the molecular structure of glucose and thus will not recognize glucose 6-phosphate, the product of phosphorylation. It is also important to point out that this step is irreversible, meaning that this glucose molecule is now trapped in the cell. Hexokinase is the name of the enzyme that phosphorylates glucose in most cells, with a low Km, meaning it functions most efficiently at a low concentration of glucose. And this makes sense because a low Km value means that it reaches half maximum velocity at a lower concentration of its substrate, glucose, which means that hexokinase functions well even when glucose concentration is low, ensuring that glucose is efficiently utilized for energy production or stored as glycogen. Hexokinase is inhibited by glucose 6-phosphate, which means that the enzyme's product inhibits it. On the other hand, glucokinase is the enzyme that phosphorylates glucose in the liver and pancreatic beta cells. It has a high Km value, meaning it has a lower affinity for glucose compared to hexokinase. This high Km value allows glucokinase to become active only when glucose levels are high, such as after a meal or when blood glucose levels rise. In the liver, glucokinase is induced by insulin and hepatocytes. Now we can start glycolysis. Phosphofructokinase 1, or PFK1 for short, is the rate-limiting step in glycolysis and functions as the main point of regulation. This step commits glucose to further breakdown in glycolysis. Again, the kinase in the enzyme's name should be a hint that our substrate is being phosphorylated again. This step is a part of the energy investment stage in glycolysis, where ATP is used to phosphorylate fructose 6-phosphate into fructose 1,6-bisphosphate. As I mentioned, PFK1 is highly regulated, being inhibited by ATP and citrate and induced by AMP. This should make sense because citrate is an intermediate in the citric acid cycle, and high levels of ATP should indicate to the cell that it can stop glycolysis because it has sufficient energy already. It also makes sense for PFK1 to be induced by AMP, since AMP is the dephosphorylated inactive form of ATP, indicating that the cell is low on energy. The difference between PFK1 and PFK2 is that while PFK1 directly controls the pace of glycolysis by catalyzing the committed step and is thus a key regulator of glucose metabolism, PFK2 indirectly regulates glycolysis by modulating the activity of PFK1 through the synthesis of fructose 2,6-bisphosphate, a powerful activator of PFK1. PFK2 is activated by insulin, which will convert a small amount of fructose 6-phosphate to fructose 2,6-bisphosphate, which in turn activates PFK1. In the other case, PFK2 is inhibited by glucagon, which reduces the amount of fructose 2,6-bisphosphate produced, thus inhibiting PFK as a result. Being largely found in the liver, the biological purpose of PFK2 is to override the inhibition of PFK1 by ATP when the cell is metabolically satisfied, and then glycolysis can continue. This is important for the liver to promote metabolic flexibility and the production of intermediates called metabolites that serve as precursors for other metabolic pathways. The next important enzyme is glyceraldehyde 3-phosphate dehydrogenase, which oxidizes the substrate and adds an inorganic phosphate group. While we know from Gen Chem that oxidation is a loss of electrons, we also learn in biochemistry that it can also mean a gain in oxygen, which is what we're going to see in this process. The products include 1,3-bisphosphoglycerate, which is a high-energy intermediate, and the reduction of NAD+ to NADH, which feeds into the electron transport chain in oxidative phosphorylation. In the next step, 3-phosphoglycerate kinase transfers this high-energy phosphate group from 1,3-bisphosphoglycerate to ADP, creating ATP and 3-phosphoglycerate. This kind of ATP formation is called substrate-level phosphorylation, as opposed to oxidative phosphorylation in the electron transport chain, which we'll see later. Pyruvate kinase is the last step in aerobic glycolysis, which also uses substrate-level phosphorylation to produce ATP using another high-energy substrate called phosphoenol pyruvate, or PEP. It is important to note that this step is irreversible, and this enzyme is induced by fructose 1,6-bisphosphate, which was produced by PFK1 earlier in the pathway. This kind of regulation is called feed-forward activation, since an earlier product is activating a later enzyme. All right. When there is no oxygen available to perform aerobic respiration, a cell will undergo fermentation, in which lactate dehydrogenase will oxidize a product of glycolysis, NADH, to NAD+, while simultaneously reducing pyruvate to lactate. This is important because if all of our NAD+ was used up in glycolysis, we would have no electron carriers left, and glycolysis would halt. Although most tissues do perform respiration aerobically, we see an accumulation of lactate in the event of intense exercise only in skeletal muscles and after a heart attack or a stroke. Some important intermediates to keep in mind from glycolysis are DHAP, DAP, 1,3-BPG, and PEP. DAP is formed from fructose 1,6-bisphosphate in the PFK1 reaction, and this product can be isomerized to glycerol 3-phosphate, which can then be converted to glycerol, which functions as the backbone in triacylglycerol synthesis, which is another important metabolic pathway. 1,3-BPG and PEP are both high-energy intermediates that can be used to generate ATP through substrate-level phosphorylation, with 4 ATP in total produced and a net 2 ATP from glycolysis overall. In the last slide, the name 1,3-BPG may sound familiar because it is almost the same as 2,3-BPG, the allosteric regulator of hemoglobin. 2,3-BPG can be produced from 1,3-BPG by an enzyme called bisphosphoglycerate mutase, which is found in red blood cells. This mutase moves the phosphate group from carbon 1 to carbon 2. We know that 2,3-BPG binds allosterically to hemoglobin, which causes a rightward shift in the binding curve, which is what we can see going on here, which represents a net unloading of oxygen in the tissues while still maintaining full oxygen saturation in the lungs. The pyruvate dehydrogenase complex is composed of a set of enzymes responsible for taking the end product of glycolysis, pyruvate, and converting it to acetyl-CoA. As we already know, acetyl-CoA is the starting substrate for the citric acid cycle, but is also the starting material in fatty acid synthesis. When there are elevated levels of insulin in our blood, it activates pyruvate dehydrogenase complexes to produce acetyl-CoA, which this product will go on to produce more ATP as well as fatty acids for long-term energy storage, but this complex is actually inhibited by acetyl-CoA. This makes sense because elevated insulin levels should indicate that the body is in a well-fed state and should focus more on energy production and storage, but the presence of end products should stop that process. At this point, pyruvate stops being converted to acetyl-CoA and instead is converted to oxaloacetate, which is the starting product for gluconeogenesis, which is our next topic. While insulin is used to lower blood glucose levels, hormones like glucagon, cortisol, growth hormone, and epinephrine raise levels of glucose in the blood, largely by starting the process of glycogenolysis and gluconeogenesis. Gluconeogenesis mainly happens in the liver to stabilize glucose levels, as well as in the kidneys to a much smaller extent. Glucagon and epinephrine both act to promote gluconeogenesis, especially in times of fasting when the blood sugar needs to be increased. Some of the substrates involved in gluconeogenesis are derived from adipose tissue, muscle tissue, and lactate from anaerobic respiration. Other kinds of dietary sugar like fructose and galactose can also be converted to glucose in the liver to be used by the body. It is important to point out that once pyruvate has been converted to acetyl-CoA by the pyruvate dehydrogenase complex, it cannot be converted back to glucose. When looking at the process of glycolysis, we can see that there are three irreversible steps, those involving the enzymes hexokinase, PFK1, and pyruvate kinase. Thus, in order for our body to bypass these steps, our body has to utilize different enzymes. The first important enzyme to consider in gluconeogenesis is pyruvate carboxylase, which is induced by acetyl-CoA. This step is important in the body's regulation of glucose levels. When acetyl-CoA levels rise, it means that the body has enough energy and need not continue the process of glycolysis. We learned that acetyl-CoA also inhibits pyruvate dehydrogenase, which is responsible for converting pyruvate into acetyl-CoA to continue the process of glycolysis. So when the levels of acetyl-CoA are elevated, it not only turns off pyruvate dehydrogenase and thus stops glycolysis, but it also turns on pyruvate carboxylase to start the process of gluconeogenesis. After pyruvate carboxylase, pyruvate is turned into oxaloacetate, which is an intermediate of the citric acid cycle that cannot escape the mitochondria. The next important enzyme is phosphoenolpyruvate carboxykinase, or PEPCK, which converts the oxaloacetate into phosphoenolpyruvate, or PEP. This enzyme is activated by cortisol and glucagon and requires GTP to operate. PEPCK in combination with pyruvate carboxylase work in junction to circumvent the irreversible pyruvate kinase step in glycolysis. Fructose 1,6-bisphosphate is the next important enzyme in gluconeogenesis and is responsible for bypassing the irreversible PFK1 step, which is the rate-limiting step in glycolysis. It does this by removing the phosphate on carbon 1 of fructose 1,6-bisphosphate to form fructose 6-phosphate. Something that may help you remember what this enzyme does is that phosphatases reverse the action of a kinase, removing a phosphate group. This particular enzyme is inhibited by AMP and activated by ATP, which makes sense since we should turn off this enzyme when energy levels are low, and we should induce this enzyme when usable energy levels are high, meaning that we don't need to keep burning glucose. Glucose 6-phosphatase is an enzyme only found in the lumen of the endoplasmic reticulum of liver cells, and as the name suggests, it removes a phosphate from glucose 6-phosphate to create glucose, which can then move out of the ER and be transported out of the liver cells in the bloodstream by GLUT transporters. It is important to note that we don't have these enzymes in our skeletal muscle because that muscle is not responsible for releasing sugar into the blood. It needs the glucose to perform its own functions instead. Overall, gluconeogenesis requires a net expenditure of energy to get from pyruvate to glucose. This energy for the process is provided by the beta oxidation of fatty acids, the resultant product being acetyl-CoA. This acetyl-CoA then goes and inhibits pyruvate dehydrogenase and activates pyruvate carboxylase, thus preventing glycolysis from continuing so gluconeogenesis can occur. The same acetyl-CoA that is produced in cellular respiration cannot be used as the source of acetyl-CoA for gluconeogenesis, since going through the process of glycolysis to get acetyl-CoA would pretty much just burn off all the glucose we just made in gluconeogenesis. Thus, the breakdown of fatty acids is essential for gluconeogenesis, which can be sourced from adipose or fat tissue during times of low blood sugar. Alright, glycogenesis is the process of glycogen, while the production of glycogen, while glycogenolysis is the breakdown of glycogen. Most of this occurs in the liver and skeletal muscle, but some of it does occur in small amounts in other tissues. The usage of glycogen in the liver and skeletal muscle differs though. In the liver, glycogen is important in regulating blood sugar levels, but in skeletal muscle, glycogen is used for energy when the muscles are being used in times of intense exercise or exertion. There are two main enzymes involved in glycolysis: glycogen synthase and branching enzyme. Glycogen synthase is the rate-limiting step in glycogenesis and is induced by insulin in both the liver and the muscle, which indicates that the body is in a well-fed state and is not actively needing glucose. Glucose synthase creates an alpha-1,4 glycosidic linkage between glucose molecules, and then branching enzyme comes in and is going to branch these molecules with alpha-1,6 glycosidic bonds. The purpose of creating branches is for rapid energy mobilization. If the body reaches fasting state and needs glucose quickly, it is much quicker to be able to break off multiple glucose molecules at the same time, as opposed to one molecule at a time with a linear structure. Similar to glycogenesis, glycogenolysis also has two main enzymes: glycogen phosphorylase and debranching enzyme. Glycogen phosphorylase removes single glucose 1-phosphate molecules by breaking its alpha-1,4 glycosidic bonds. In the liver, this enzyme is induced by glucagon, indicating that blood sugar is low and glucose is needed, and in skeletal muscle it is induced by AMP and epinephrine, both of which indicate a need for usable energy in the muscles. Lastly, debranching enzyme is able to move whole oligoglucose sections at a time from one branch to the chain, utilizing alpha-1,4 glycosidic bonds while simultaneously removing a branch point, releasing one glucose. The pentose phosphate pathway, also known as the hexose monophosphate shunt, is a type of fuel storage pathway that occurs in the cytoplasm of all cells. It has two main functions: producing NADPH and ribose 5-phosphate. NADPH is an important electron donor and thus reducing agent in a number of biochemical reactions, while ribose 5-phosphate is used in nucleotide synthesis in the cell. The main important enzyme in this pathway is glucose-6-phosphate dehydrogenase, or G6PD. It is the rate-limiting step of this process and is activated by insulin, which makes sense because this is a fuel storage pathway. Now, let's take a closer look at acetyl-CoA. This substrate enters the citric acid cycle and is oxidized to CO2 and water, creating the high-energy carriers NADH and FADH2. Along the way, that will be later used in the electron transport chain. As a review, we know that after glycolysis, its product pyruvate will enter the mitochondria through active transport to be oxidized and carboxylated by pyruvate dehydrogenase complex. In this irreversible reaction, we will be going from 3-carbon pyruvate to 2-carbon acetyl-CoA and CO2. Due to the irreversible nature of this reaction, we cannot work backwards from acetyl-CoA to form glucose, and instead we start with pyruvate. The reaction converting pyruvate to acetyl-CoA is highly exergonic with a delta G of -33.4 and is inhibited by the accumulation of NADH or acetyl-CoA, since both indicate that the electron transport chain may not be functioning properly. The first enzyme found in the pyruvate dehydrogenase complex is pyruvate dehydrogenase, or PDH, and it is responsible for oxidizing pyruvate, producing CO2 as a side product. The resulting 2-carbon molecule is covalently bound to thiamine pyrophosphate, which is vitamin B1 or TPP, and magnesium is also required for this step. TPP is a coenzyme that can be found non-covalently bound to PDH. Next, dihydrolipoyl transacetylase is responsible for oxidizing the 2-carbon molecule attached to TPP and transferring it to lipoic acid. Lipoic acid is a coenzyme that is covalently bound to dihydrolipoyl transacetylase and contains a disulfide group which acts as a great oxidizing agent, catalyzing the creation of the acetyl group. The enzyme then transfers this acetyl group to CoA, forming acetyl-CoA. After this, lipoic acid is left in a reduced state and needs to be oxidized to be functional again. In the next step, FAD is a coenzyme that will come in and reoxidize lipoic acid, restoring it to its original state. In the process of oxidizing lipoic acid, FAD itself is reduced to FADH2. In later reactions, FADH2 will be used to reduce NAD+ to NADH. Pyruvate dehydrogenase kinase and pyruvate dehydrogenase phosphatase are both enzymes used to control the activity of PDH, with pyruvate dehydrogenase kinase turning off PDH when ATP or acetyl-CoA levels are high by phosphorylating it, or conversely, pyruvate dehydrogenase phosphatase dephosphorylates PDH when ADP levels are high, turning it on. In this case, we can use the names kinase and phosphatase to guess what these enzymes do. The effect of phosphorylation is opposite of what we would expect, as it turns off the enzyme rather than turning it on. It is important to note that while the pyruvate dehydrogenase complex is the main producer of acetyl-CoA that we think of, there are other methods for making the substrate to be used in the citric acid cycle. In fatty acid oxidation, a thioester bond will form between the carboxyl group of fatty acids and CoA bonded to a thiol group. This process is called activation and produces acyl-CoA, which cannot cross into the mitochondria on its own. Instead, fatty acids will enter the mitochondria using a carrier called carnitine. This forms a complex called acylcarnitine, which will transfer its acyl group to a mitochondrial CoA group to reform fatty acyl-CoA, which can undergo beta oxidation to form acetyl-CoA. Acetyl-CoA can also be formed from the carbon skeletons of ketogenic amino acids, ketone bodies, and alcohol. The next step in aerobic respiration is the citric acid cycle, which takes place in the mitochondrial matrix. Although oxygen is never directly used as a substrate in this process, the citric acid cycle cannot occur anaerobically, since a lack of oxygen would prevent the electron transport chain from occurring and consuming NADH and FADH2, which would lead to the accumulation of these electron carriers in the mitochondria. We will later talk about the fact that NADH and FADH2 both serve as allosteric inhibitors of the enzymes used in the citric acid cycle. The net products of the cycle per turn are 1 ATP, 3 NADH, and 1 FADH2. I bolded per turn because one glucose molecule will correspond to 2 turns of the citric acid cycle, since each 6-carbon glucose molecule is turned into 2 3-carbon pyruvate molecules in glycolysis. So the net products per glucose molecule would be doubled. As we go into the next slides, there's going to be a lot of substrates to remember for the MCAT. So to help me remember all of the substrates in their order, I like the mnemonic that I've put here, which is, "Can I keep selling seashells for money, officer?" So you'll see that in the next slide. Okay, so in the first step of the citric acid cycle, acetyl-CoA and oxaloacetate are going to come together through a Claisen condensation to form the intermediate citral-CoA. A Claisen condensation is just when you use water to form a new bond between 2 carbonyl compounds at the alpha carbon. The intermediate is going to be hydrolyzed to form citrate. The enzyme used in this step is citrate synthase, and something helpful to remember is that synthases are enzymes that create covalent bonds between molecules without much energy input. In step 2, we start with citrate, which is a tertiary alcohol. We want to oxidize our substrate, but tertiary alcohols cannot be oxidized to carbonyls without breaking the octet rule. So instead we will use an enzyme called aconitase to isomerize citrate to a secondary alcohol intermediate called cis-aconitate. To go from citrate to cis-aconitate, we must lose water, but to go from our intermediate to isocitrate, aconitase must add water back to form isocitrate, a secondary alcohol. Although this reaction is in chemical equilibrium, in order to push this process forward towards our desired product isocitrate, the quick consumption of isocitrate in the next step will drive this reaction forward to replace the lost isocitrate. In the third step, the oxidative decarboxylation of isocitrate is catalyzed by isocitrate dehydrogenase. What this means is that the carboxyl group highlighted in green here will be removed and the OH group will be oxidized to a carbonyl, with the hydride from that OH group being transferred to NAD+ to form our first electron carrier, NADH. It is super important to remember that this step is the rate-limiting step. One trick that may help you remember the product of this reaction, alpha-ketoglutarate, is that if you were to replace the carbonyl group here with an amino group, you would be left with the amino acid glutamate, which has a very similar name. Both of these steps involve dehydrogenase enzymes, which are a subtype of the oxidoreductase class of enzymes responsible for oxidation reduction reactions. In the case of dehydrogenases, these enzymes specifically transfer a hydride ion to an electron acceptor like NAD+ or FAD. The fourth step involves another oxidative decarboxylation, but this time the mechanism is going to be different, and instead resembles the process of pyruvate dehydrogenase oxidating pyruvate to acetate. We're going to net another NADH and CO2 from this process since we're losing a carbon and a hydride. We're left with succinyl-CoA, which has a high-energy thioester bond between the sulfur and the CoA part, which will become super important in the next step. In step 5, the high-energy thioester bond is hydrolyzed, releasing a decent amount of energy that is coupled with GTP synthesis. GTP is another high-energy molecule like ATP that is involved in substrate-level phosphorylation. Later, this GTP can be used to phosphorylate an ADP molecule to produce ATP. The enzyme in this step is a synthetase, meaning it creates covalent bond with the input of energy. It's important not to mix these up with synthases, which do not require the input of energy to create covalent bonds. Now that we have the molecule succinate, we know that our end product of oxaloacetate also is a 4-carbon molecule that only differs by having a ketone as opposed to the methylene that we have right here found in succinate. We can work towards making oxaloacetate by oxidizing the beta carbon, or the carbon next to the carboxyl group, also the methylene. Since the molecule is symmetrical, it doesn't matter which one we do. This conversion of succinate to fumarate is the only reaction in the citric acid cycle that does not occur in the mitochondrial matrix, and instead occurs on the inner membrane of the mitochondria. Since the enzyme used, succinate dehydrogenase, is an integral protein in the inner mitochondrial membrane, it is also covalently bound to the electron acceptor FAD, or flavin-adenine dinucleotide, making the enzyme a flavoprotein. We use FAD instead of the usual electron acceptor NAD+, because succinate actually does not have enough reducing power to reduce NAD+. Step 7 is actually super simple, and fumarate is simply going to hydrate our alkene substrate fumarate to form malate. Although technically either enantiomer could form from this reaction, only L-malate is going to be formed. In step 8, malate dehydrogenase is going to use NAD+ to oxidize malate to oxaloacetate. This step is important because we have now regenerated our starting product of the citric acid cycle, hence why it is called a cycle. Now the cycle can repeat again. There are 3 main control points in the citric acid cycle to know. The first occurs with citrate synthase. ATP, NADH, succinyl-CoA, and citrate, the product it generates, act as allosteric inhibitors. These inhibitors signal high energy levels and abundance, resulting in a slowdown of the cycle's pace. This regulatory mechanism ensures that when energy levels are high, the cycle's activity decreases to prevent unnecessary energy expenditure. The next control point is isocitrate dehydrogenase, which converts isocitrate to alpha-ketoglutarate. ATP and NADH act as allosteric inhibitors, indicating high energy and electron carrier abundance, thereby slowing down the cycle. Conversely, ADP and NAD+ act as allosteric activators, signaling low energy and electron carrier levels, promoting cycle activity. Lastly, the alpha-ketoglutarate dehydrogenase complex converts alpha-ketoglutarate to succinyl-CoA. Its activity is regulated by different molecules. ATP, succinyl-CoA, which is its product, and NADH act as allosteric inhibitors, signaling ample energy and reducing power, thus slowing down the cycle. ADP and calcium ion act as allosteric activators, indicating low energy and increased calcium levels, producing cycle activity. Regulation of these enzymes ensures optimal operation of the citric acid cycle, responding to cellular energy needs and maintaining a metabolic balance. Lastly, we're going to look at the electron transport chain, which is the final step of aerobic respiration, where it takes all of the electrons it's harvested from different sources and converts it into usable energy for the body. The goal of the electron transport chain is to couple the exergonic electron transport process with the endergonic process of synthesizing ATP. While the citric acid cycle occurs in the mitochondrial matrix, the electron transport chain occurs on the matrix-facing surface of the inner mitochondrial membrane. This inner membrane is characterized by having folds in it called cristae, which serve the function of maximizing the surface area over which electron transport can occur, thus maximizing energy production. The electrons in the name electron transport chain come from NADH, and these electrons are transported using redox reactions, in which the reduction potentials gradually increase until reaching oxygen, the final electron acceptor. While the processes of electron transport along the mitochondrial membrane and ATP formation are technically separate processes, they are highly coupled. Because what happens in electron transport is that the high-energy electron carriers, NADH and FADH2, are going to transfer their electrons to specialized carrier proteins that will begin a cascade of electron transfer dictated by the reduction potentials, eventually leading to the final electron acceptor O2, which has the highest reduction potential of all. As we remember, the direction of electron flow in oxidation-reduction reactions is determined by the reduction potentials, and the molecule with the highest reduction potential will be reduced while the other is going to be oxidized. The electron transport chain is simply a series of oxidation-reduction reactions that have increasingly high reduction potentials until reaching oxygen with the highest. The first transfer of electrons is between NADH and coenzyme Q. NADH is a great electron donor and will donate these electrons to a flavoprotein with a coenzyme called flavin mononucleotide, or FMN, which is covalently bound. Now, NADH is oxidized to NAD+ as FMN is reduced to FMNH2. Another redox reaction occurs when the flavoprotein is reoxidized while the iron-sulfur subunit is reduced. Now, the iron-sulfur cluster can donate these electrons from FMNH2 to coenzyme Q, which is also called ubiquinone. Now, coenzyme Q becomes CoQH2, which this is an exergonic process. As these electrons are flowing through complex 1, they are losing energy, which is harnessed to pump 4 hydrogen ions from the matrix to the intermembrane space, starting the proton gradient across the mitochondrial membrane, which will be essential later for ATP synthesis. In complex 2, FAD is covalently bound and is going to be reduced by succinate to form fumarate and FADH2. We may remember that succinate is an intermediate in the citric acid cycle that gets oxidized to fumarate while reducing FAD, and the reaction is catalyzed by succinate dehydrogenase. A similar mechanism will occur here at complex 2. FADH2 will go on to pass its electrons to the iron-sulfur cluster, which will in turn be reoxidized by transferring its electrons to coenzyme Q, forming CoQH2, similar to what happens in complex 1. Complex 3 is also called cytochrome reductase because it facilitates the transfer of electrons from coenzyme Q to cytochrome C. Cytochrome C is a type of protein that contains a heme group that can be oxidized from Fe2+ to Fe3+. Since coenzyme Q has 2 electrons to donate, but cytochrome C can only take 1, 2 cytochrome C molecules must be used. Although the drawing above depicts 2 separate complexes, there's really only one, and it was only drawn this way to illustrate the 2 separate redox reactions going on. This electron transfer is coupled with the transfer of 4 protons from the matrix to the intermembrane space through a process called the Q cycle. It is important to note that neither coenzyme Q nor cytochrome C are actually part of the complexes being described, but rather are freely moving within the membrane, facilitating transfer from one complex to the next. Lastly, complex 4, also called cytochrome C oxidase, is responsible for transferring electrons to the final electron carrier, oxygen. Copper is an important cofactor in the process, which makes up one of the subunits in this complex along with cytochromes A and A3, together called cytochrome oxidase. Cytochrome oxidase is oxidized in a series of redox reactions as oxygen becomes reduced to form water. This is the last location of proton pumping in the electron transport chain, and a total of 2 hydrogens are pumped over the membrane in this step. So in 1 molecule of glucose, the net ATP produced in aerobic respiration is between 30 to 32 ATP. The reason for this discrepancy depends on the NADH shuttle mechanism used. NADH is a high-energy electron carrier, as earlier discussed, but it's actually incapable of crossing the inner mitochondrial membrane to contribute its electrons directly. There are 2 shuttle mechanisms that facilitate this transfer, and depending on which method is used by the cell, either 1.5 or 2.5 ATP will end up being synthesized, accounting for the discrepancy. In the glycerol 3-phosphate shuttle, NADH transfers its electrons to DHAP DAP, forming glycerol 3-phosphate. These electrons can now be used to reduce mitochondrial FAD to FADH2, which will in turn transfer its electrons to the electron transport chain via complex 2, leading to the synthesis of 1.5 ATP for every NADH molecule. On the other hand, malate aspartate shuttle uses an enzyme called malate dehydrogenase, which transfers the NADH's electrons to oxaloacetate, forming malate. Since neither NADH nor oxaloacetate can cross the inner mitochondrial membrane, instead, transfer to malate in the cytoplasm is needed to cross the membrane. Once malate crosses the membrane, malate dehydrogenase will reverse the reaction that just occurred, reforming NADH. Now that NADH is inside the matrix, it will contribute to the electron transport chain by transferring its electrons to complex 1 and will go on to produce 2.5 ATP per NADH. In order to refresh oxaloacetate for future shuttles, malate is oxidized to oxaloacetate and transaminated to aspartate, which is able to cross the membrane back to the cytosol. Then, aspartate is turned back into oxaloacetate and the cycle is able to repeat. Lastly, in oxidative phosphorylation, the chemiosmotic gradient created by the high concentration of hydrogen ions in the intermembrane space and low concentration of hydrogen ions in the matrix provides the energy needed to complete the intergonic process of phosphorylating ATP to form ATP. ATP synthase has 2 different portions, the F0 and the F1 portion. The F0 portion is where the hydrogen ions flow through down their gradient, which releases the chemical energy harnessed by the F1 portion of ATP synthase, which is responsible for phosphorylating ADP. Remember that we discussed during the citric acid cycle that synthases are enzymes that do not require the input of ATP or to form their covalent bonds, and the bond in question here being an inorganic phosphate group and ADP. Respiratory control is the coordinated regulation of oxidative phosphorylation. The citric acid cycle is inextricably linked to ATP synthesis, meaning that in times of low oxygen, the rate of oxidative phosphorylation will decrease, leading to an accumulation of NADH and FADH2, which are both inhibitors of the citric acid cycle. This way, oxidative phosphorylation works together to meet the cell's needs. And this is the end of metabolism, and thank you so much for watching. Have a great rest of your day.